So I guess it is 5 o'clock or the hour now. So I guess we can get started. So as you might have noticed, this is a dialogue. So it's just gonna be kind of like a conversation between us two. And I thought we could start off with by introducing ourselves. So, Arula, do you want to introduce yourself?
Sure. So my name's Arula Ratnakar. I'm a neuroscientist and science fiction writer. You can find my four published stories in Clarkesworld magazine. My most recently published story came out in February called Babarusa, and I tend to write all about like neuroscience stuff I'm interested in, biology stuff I'm interested in, and so on.
Cool. And I'm Andrea. I'm a research fellow at a lab at Harvard, which studies human genetics and neurodevelopment. However, it is a new field for me, so I'm just beginning to appreciate the amazingness of the human brain. And I'm also a science fiction author, of course, and you can find my stories in Clarkesworld, Lightspeed, and Fireside, among other places. Um, Cool. And I also wanted to say we hope there will be audience participation on this event, so please feel free to put comments, questions, anything. I have eye on both Discord and in the chat, so whatever you prefer, and hopefully we'll incorporate that into the dialogue. All right. So in general, I thought we would follow an outline where like the first half is broadly about misconceptions, and then the second half is about exciting advances in biology. So I guess we could start off with misconceptions, and I guess a good place to start might just be like portrayal of biological sciences in popular media. And of course, we had one of those mentioned in the panel description, which is No Time to Die. So did you actually see that movie, Arula?
So I started watching it right before this panel, but I wasn't able to finish it in time. But I did get to the like part where they talked about like the nanobots, I think entering
Perfect.
the bloodstream.
So I guess you could provide our first reaction of a biologist to the villain in No Time to Die.
Um yeah, so I guess interesting portrayal of a scientist for sure. They always are though. Like I don't know. The way that science and like scientists are portrayed in media is very interesting. I don't think like the sort of like lone genius sort of thing is accurate.
Yeah, and I was willing to go for it. Like I was excited to see like CRISPR. It wasn't okay, they didn't actually say CRISPR, but it was like almost CRISPR. So I was like, willing to like be invested in it, if they had like a good, thoughtful idea of CRISPR being used in like such a villainous way. I guess if people don't know, basically this villain uses like this gene targeting virus to like kill specific individuals and people who are like genetically related to the oh sorry, I guess I should have said spoiler. Uh
Yeah.
Sorry about that. Yeah, so anyways, like they basically use this they don't mention it, but it's basically based on a real gene editing technology called CRISPR, and I was like interested in seeing them build upon that. But then they just go on to use it in like such like an unrealistic way. That I was just like there were real concerns about like gene editing. You don't have to like go like on this totally crazy tangent.
Yeah.
I guess someone in the chat said you have to take the bond factor into account. Well, okay, I have a whole rant about this, but I also feel like they had a huge missed opportunity for a DNA themed villain, right? Because like this guy is not like he uses DNA, but it's not like his evil villain theme at all, right? It's just like shoehorned in. It's like someone read one article about like CRISPR, and then they were
Oh
like,
my
"Oh, let's
gosh.
put
Yeah.
this in the movie," and it's like no relation to the other parts of the movie.
Yeah, no, I mean, CRISPR is like a fascinating technology. I feel like it's always a missed opportunity to like really like get people excited about science and how it works for sure.
Yeah, and I guess so. Just to make it clear, there is a big misconception in how CRISPR, and it's spelled CRISPR, how it's used in the movie, which is that it can be used to target like literally one specific person, like have a personalized virus, which will only kill that person. That just built on so many misconceptions. And the big one, which is my pet peeve, which is like that we actually know enough about the genome to be able to you know, target someone to kill them based on their differences with like another human being.
Yeah, no. There's so many factors involved. There's like methylation. There's like, and there's like just it's yeah, no,
Yeah.
definitely.
Yeah, and I guess so the takeaway is that there's a lot about human traits which are not, defined based solely on DNA. So Rula mentioned something called methylation, which is a layer of regulation, which is known as epigenetics. So this is like a layer of regulation on top of the DNA sequence itself, and this is actually, incredibly important for like differentiating individuals. And like by definition, it's not something that can be targeted from the DNA sequence. Yes, I will spell methylation.
Yeah, basically, you can sort of like silence or like if something is methylated, if a portion of the genome is methylated and that it's like a sort of chemical group called a methyl group is added to some part of it, then it can be like silenced or activated in like different ways.
Yes, and just also to um, scale back a little bit. So the part of the so we have 3 billion base pairs in the human genome, and everyone loves to focus on genes, which you always hear in the news like, oh, genes just they decide who you are. You get these genes, that's it. But that's totally not true because actually, so only 1% of the human genome is consisting of DNA that actually codes for like things like proteins and enzymes that carry out like functions in the cell. And the other 99% of the genome, like, we know very little about how it actually works. And we think, I mean, part of the answer is in this regulatory function. So, like, regulating how, the genes are expressed, which is also related to epigenetics.
I see in the chat that someone wanted to know about telomeres, so I can talk. So so in eukaryotic cells, which are basically cells which have their like genomic DNA housed within this structure called a nucleus, chromosomes, which have the DNA, have these sort of caps on the ends called telomeres, which contain sort of hundreds or thousands of repeats of the same short DNA sequence. Um, and they kind of need to be protected from DNA repair systems because they have these overhangs that are single stranded and our DNA is double stranded. And this triggers like DNA repair systems to like, or like this basically makes it looks like damaged DNA that needs to be like addressed. And this is because of like incompleteness in replication of the DNA. And every time it's replicated, it's sort of like there's a little bit that's like left incomplete. So it kind of so proteins associated with like telomere ends kind of protect them from triggering DNA repair pathways, but they can like but basically they get sort of like shorter and shorter over time. I don't know if that was a good explanation.
It was great. So I assume the reason why this was asked about is because of the conception, which you also see in a lot of popular media, that basically telomeres, control longevity. So basically, if you had infinitely long telomeres, you would live forever. So I don't know. Do you want to? What is your hot take on that, Arula? That
Um,
conception.
Well, there's some like mouse and rat studies that kind of show that kind of reversing telomere shortening via like I don't know. There. I think obviously nothing has been done, and like it shouldn't be done in humans right now. But like, I think from what I remember about like the some animal studies, they like they kind of reversed telomere shortening by binding to like um uh, by having the telomeres bind to like a special RNA molecule that added some nucleotides to the overhanging strand of the telomere DNA using the complementary RNA as a template. And then a matching strand was made by like the normal DNA replication machinery producing like double-stranded DNA. So it just increased the length and like and I think that they did show that it kind of like slowed down the aging and the mice or rats that underwent this treatment. By that they just meant like behavioral sort of analyses of like they were like more active, they were like grooming and stuff, and they weren't showing like the signs of aging as much. But I think it's an extremely just like early. I don't think we can make any solid conclusions yet.
Mm.
There are definitely like, but I'm also not an expert in this field.
Yeah, well, I think like the important takeaway though is from the research that has been done is that it's definitely not sufficient to like, extend someone's life or an organism's life indefinitely because yeah, we have laboratory mice that have like gigantic like telomeres that are just like actually I think even normal strains of lab mice tend to have like very long telomeres. And obviously these mice are not living forever, so you can't just make someone live forever by giving them infinite telomeres. So I think it's probably more like it's, necessary if you want to live for a long time to have long telomeres because like if the telomeres if you don't have telomeres then like yeah, DNA replication is just going to consistently like be chopping down your DNA and obviously that's bad for living. Um but yeah, it doesn't seem like it's like the slam dunk to living forever just to have like super long telomeres and there's still ongoing research. So
Yeah, and in terms of like, I mean, I feel like telomeres are talked about all the time, but one thing that I wish was talked about more is like the aging of the immune system, which I think is for in terms of the whole like extending lifespan and stuff like that. One fascinating study I read about was where so we have this organ called the thymus, which is basically where like T cells for our immune system are like sort of raised, and they turn from like baby T cells to like T cells that are like ready to go out into the world. And like children and like young sort of people have thymuses that are generating like lots and lots and lots of T cells all the time. But then as we age, the structure of the thymus is like sort of infiltrated by fat tissue, by like fat cells, and like and like fewer and fewer T cells are produced. So they did this study where they like put a young rat's
oh yeah.
thymus into an old rat, and they found that like the age of the rat was just like, I mean, behaviorally it was just acting young again. It was fascinating. So there's so many other factors to aging beyond telomeres, that there's so many different like avenues by which we could like explore like sort of extending lifespan and that whole science too.
Yeah, definitely. I think the immune system in general is just like very well, people are definitely studying it, but I think people are just beginning to like appreciate that the immune system, it doesn't just fight off viruses and infections, but it like plays a huge impact on basically, you know, every aspect of our body and our lives by extension. And yeah, there's even like a lot of work on the impact on the brain, of course, which is also extremely interesting. Um, so should we take some more questions, I guess, from the chat? Or I guess Arula, since I went off on my rant, is there any like one myth about, anything bio related that you just like to bust right now in front of our audience?
Oh yeah, yeah. So one, I mean, okay, I mean, I work with animal model organisms, so I might, have like some personal opinions on this because it is like what my research is in. But I there's this sort of argument I hear often that like, oh, like why do like mouse or rat or like zebrafinch brains like matter if we're trying to understand the human nervous system? And like that's a myth I'd like to bust because there's so first of all, like we're very limited in the number of ways we can sort of like ethically perturb like human brains to understand their mechanisms. Whereas in like mice or rats, you have all of these tools at your disposal, such as like optogenetics, multiphoton microscopy, all sorts of stuff that you can use to like literally in real time visualize the neurons or like stimulate neurons and like see behavioral results and like do or like you can do so much more with animal model organisms. That's one thing. Also, there's several conserved structures across like species, like the basal ganglia, for example, is super similar in humans and in rodents. The hippocampus is like there's similar structures that are conserved, and there are also like several tasks that behaviorally humans and the animal model organisms perform similarly on. And so if you're looking at the conserved structures between humans and those model organisms within those tasks and the frameworks of those tasks, you can begin to answer questions about the brain. And also the final thing is that so someone in like a lab in the building that I work in, Dr. Steve Ramirez's lab, he has this saying he talks about whenever this argument is brought up, where like a tricycle and a Lamborghini are not the same thing, but you can understand mechanisms of a Lamborghini by studying a tricycle. And so that's kind of, yeah. Anyway, those are my thoughts on why studying animal model organisms is really important for neuroscience research.
Yeah, those are great points. Yeah, and also on the molecular level, like the genes that are very important for neurodevelopment, they've been very conserved throughout throughout mammals. So even especially at the molecular level, having like such strong like genetic manipulation tools in mouse models, that also is also very relevant, can be very relevant to even study like the molecular side, neurobio. All right, so we have some interesting questions in the chat. So one of the questions is about Kingsman, which I haven't watched. Have you watched that?
I haven't seen it yet.
Okay, so I guess we'll just have like a very broad overview of this question, probably. Uh, so sounds that can drive you crazy or irrationally angry, is that based on anything at all?
Sounds that can drive like just
Make you very angry?
like sensitivity to sounds.
Yeah, I think that's the broad concept probably that they built on in that movie.
Oh, I see. I mean, if there were parts of like the auditory processing system that were especially sensitive, then I'm sure that would. Oh, that's interesting. I mean, like the nice thing about science is that you can't like okay, there's science for science and then there's science for science fiction, and I think science for science fiction is this amazing creative space where you can just take a bunch of science and use it to like imagine whatever you want. So yeah, I'm sure there's like a way we can come up with like how using neuroscience you can get a sound to like but it would be like it wouldn't be like feasible or allowed.
Right. Yeah,
Science fiction science.
yeah, I think that's a really good point. So I think definitely both doing like actual research science and science fiction, like they both require creativity. And it's just like the way that creativity is like channeled that turns into like science fiction versus like research. Because when I come up with an idea, as Rula mentioned, like if we come up with the idea, like what about a sound that makes people really angry? If we are in a research lab, well, first of all, we need to think about the plausibility of addressing that question in research, which you're definitely no one's getting funded for that, so not happening. Um, and then like if even if you were to undertake such a project and in most cases, actually, most hypotheses you end up coming up with negative data, and in actual scientific research, it's just like, well, yes, the hypothesis is out the window. But the amazing thing about science fiction is that even if it's not supported by experiments in real life, you can still write like a science fiction story about it. That's like the amazing thing about science fiction.
Yeah, I like to think of them as like I when I'm talking to people about writing science fiction, it's like you can use the research to come up with like hypothesis chains, I call them. Like if this hypothesis were true, what would be the set of like results? And if one of those were true, and then you keep doing that until you've created like a whole future. And it's like an amazing way to like sort of it's part of why I really love writing science fiction. It's like a way to interrogate research in a way that you can't do using actual research. You can do it in science fiction at a much faster pace, at a much like you have much more like um freedom to sort of philosophically and even like engage with these scientific ideas and just like come up with like wow, what is like the furthest you can take this cool science idea.
Yeah, that's such a cool way of thinking about it. And yeah, this probably plays into another question from the audience, which is advice for like keeping the speculative science elements realistic without overwhelming readers, and like basically how do you find your own balancing act?
I mean, I think I actually still kind of struggle with this. I feel like sometimes I put too much science into my stories, but like I don't know. I don't actually really mind it that much either. But it's definitely an exercise in okay, well, I guess one thing I can say is don't make the science like don't ever I think if you get yourself like excited by the science and you're like, I just want to communicate how I want to communicate and like make accessible to everyone how exciting and interesting the scientific idea is, then I think you will have an easier time weaving it into a story rather than, oh, there's this dense chunk of science. How do I like add it to the story? Like if it's like, how do I use this story to communicate the science and make it accessible? Then it wouldn't feel as like dense and like boring or jargony.
Yeah, I definitely yeah, I definitely agree with everything you said. But yeah, I've also found I'm still also on this balancing act, still struggling to find balance. And it's basically I found so far, at least for like my direct research, it's basically really hard to like write science fiction about it. I think because yeah, like you said, it's really hard to differentiate between like an actual like research paper and like turning it into fiction. So yeah, something I've definitely found as a scientist is just like writing something that's like at least like kind of one step removed from like my actual literal research. So I mean, if there's any scientists or science loving people out in the audience, also on the balancing act, um, yeah, one thing I would suggest is like sometimes you think the easiest thing to write about is actually not that easy to write about because the closer it is to you, like the more high standards you have for like getting the science right. Um, so yeah, I think a part of it that I'm trying to do now is just like be patient with myself and I like for example if I want to really write about a science fiction story about epigenetics, but now it's turning too much into an epigenetics paper, then I'm just like taking one step back from it and maybe writing about different kind of science which I don't directly do research in, but I'm still very interested in. Okay, this is a fun question. Real life bio things that are super cool, weird, interesting, that it blows your mind that they're not used in fiction more.
Oh, I mean, Optogenetics is what I wrote a novella. I mean, Submergence. You can read it in Clarksville. That was basically like optogenetics is so cool. It's just like put it in humans in the science fiction story because it's such a cool technology. Basically, what it is I mean, like, if I were to summarize it in one sentence, it's controlling a neuron with light. But basically, you genetically modify neurons to contain this light sensitive ion channel. So, I mean, the way that neurons like send their electrical signals is through like the opening of ion channels that allow, like, positively charged ions to like flow in, and that causes the electrical spike. So if you add this thing called a light gated ion channel into the neuron's membrane, it's normally found in algae, but if you take that protein and then you like put it into the neuron membrane, then when you shine a light on it, it will actually open and allow ions to flow through, so you can like turn on the light and cause the neuron to like fire and the action potential to like sort of spike. So we use this in like memory neuroscience, for example, to sort of like reactivate cells that were like tagged to have been active during a specific experience. And if you like reactivate that group of cells, you see that they display the same behavior they did during when they were back having that original experience. So it's like you're reactivating sort of the memory, but that's kind of, it's more like the cells associated with that experience, but via light. So I mean, it's so science fiction-y.
Yeah, I know. No, alpha is so cool. And yeah, I really admire like how you used it in Submergence, and yeah, I just love it in your work whenever you use like a real like G name or something. I'm like, yes, so cool. And I guess on my side, I guess something that is already used in fiction, but I kind of wish it was used in a certain way more, is just like genome engineering. Because in my mind, like we're already almost at, well, we're basically in a science fiction world where basically because the CRISPR system is so easy to use, and basically we can order whatever, DNA sequence we want from companies. I mean, hopefully they have some controls on that, but theoretically, yeah, people could basically be, trying genome engineering in their own garage. And yeah, there are reports of like people doing this already. Um, and like there was even back in 2020, there was an article about MIT researchers making like their own like homemade COVID vaccine, which wasn't, that wasn't, I guess, gene engineering, but basically it's just like we're at this stage where people can like do it yourself, biohacks. And it is an idea that appears in fiction, but mostly in like a dystopian way. So I am just really eager to, maybe I just haven't looked in the right place, but I'm really eager to like read fiction about, people doing that in a positive way. And I would say I have tried to write about myself, but again, it's like running into that where I know there's a lot of obstacles to it, basically, mostly in like delivery of like the gene editing reagents. So I find it a bit difficult to personally write about, because like, oh man, people would never be able to do this. But yeah, I would love to like yeah, read more fiction, like biohacks for good. Yes, it is very interesting and terrifying. It's a good description. So someone in the chat wrote that's interesting and terrifying. Yeah, I guess so. Someone in the chat wrote like the sequences for like diseases like smallpox and 1918 flu are available online, and does that mean people could literally do it in their garage? So it's definitely a fear, and actually, I when I took bioethics, this was something they discussed a lot, which is like, is it ethical to publish these sequences? And of course, now we have so many like COVID research papers. So like the COVID genome has been extensively published, and that is a very important question of, as I said, like people can order like whatever DNA sequences they want, and there's some control over it. Actually, in a research lab, like for example, if you try to order like a COVID sequence from like a DNA synthesis company, they actually have filters that check for it, and they don't allow you to do it. Yeah, because this is such a fear. And doesn't mean people could literally do it in their garage. I think, it's one of those things where theoretically it's yes, but in practice it would be very difficult to, actually do, thankfully. Um, I don't know, Rule, you have any thoughts about do it yourself bio for good or evil?
I think I don't have enough expertise to comment, but like, um, I I I I don't know. Like from what I know, it's so difficult just to acquire anything and get through the paperwork to get anything approved in like a research setting that like I'm not sure what I'm not really sure what the process is to do that like outside of a research setting, but I know that everything just moves so slowly in research that I don't I don't know. And like there's so many sort of checks on what's being ordered. Like I don't know, like in the lab right now, we're gonna try and do this study looking at like psilocybin effects on like as a like a therapeutic for like chronic stress, and it's so difficult to like get psilocybin, for example. I don't know like how, yeah, I don't know. I don't think it's my area of expertise though.
Yeah, I mean, I think it's a lot. It's a question like a lot of scientists are asking, or a lot of things. It's like the same thing a lot of scientists are saying to themselves now. Like, I don't know. Like, in the research lab we have these restrictions, but if someone went rogue, like, what could they actually do? Um, yeah, and someone in the chat wrote, yeah, there's some chemicals you cannot buy without a special license or being in an academic library, and that's definitely true. But like we're getting to the point where people can like almost be able to buy machines that just like buy the machine that synthesizes the DNA on their own. So I know there's like definitely fears about that in the community. Yeah, and then someone wrote, "There's not much that would prevent me from walking out the door with it." Yeah, like as a researcher, yeah, because like as a researcher, Rolla can speak to this too, but it's not like they have a camera on us at all times.
Yeah.
So if one of us wanted to walk out with a tube, then what are the security measures in place? And most of us are asking like we don't know. You know,
Yeah,
well, most of us are saying that, yeah.
I mean, we would definitely get in major trouble, but like we could.
But
Just
yeah,
like walk out with like like the triple transgenic rat, like
we're like, yeah, what? And then sometimes, like, yeah, someone wrote basically it has happened before with a virus,
yeah.
unfortunately. So, yeah, I actually think, yeah, this is something we do need to watch out for. So, um, there's a conception that in some works of fiction that like biology labs are like ticking time bombs, and I want to say that's a misconception, but yeah, I do think yeah security protocols could be increased, but yeah then it gets into a balancing act with, well, we can't, actually at the beginning of the recombinant DNA era, like when researchers were just starting to make synthetic DNA, there was a huge like outcry over this. People were so concerned, and I think we opened that box, and we can't say to researchers, you cannot make synthetic DNA anymore. You can't say to researchers like no more like DNA research allowed. Um, so yeah, we have to. I think we could definitely increase security, and yeah, that's also out of my wheelhouse as well. Like, how would that actually look in reality?
Yeah, I mean, like thinking through, like just if someone's already approved to have access, there's like a huge process to actually get approved to work with all of these substances, with these chemicals. But once you're approved, I guess there's not much stopping you, but you would have to get, I don't know. That's kind of scary to think about, actually.
Yeah, don't panic. Please don't leave.
Yeah. No, no, no.
Let's
But
talk
like,
about positives. Um, okay, this is a good question. Like, how do you vet your biological musings for accuracy or reasonableness?
I guess, so I mean, I I write science that would be plausible. I think like based on research that I do, like for example, in Barbarossa, which just came out in February in Clark's World, I came up with this like nanotechnology where um I mean, there was some sort of like, hand wavy stuff that has to happen in order to like have this, but like I tried to like the more detailed the section was, I tried to like put like less detailed portions in the like hand wavy stuff and like more detailed portions in the plausible stuff. And that kind of made the whole thing, I think, seem plausible. Like I used actual proteins like dynamine GTPase, and like I use like graphene lattices and like the property that they have where like if you like have a stack of graphene lattices and you rotate every other one 1.1 degrees, that turns into this Mott insulator where basically based on like the energy bands you like it causes, even though it's a technically electrically conductive structure, when you rotate them 1.1 degrees, it causes like uh, it stops electrons from being able to flow through anymore. So I used that in combination with like this like rotating protein called dynamine GTPase to create this like mechanism that could like turn something from conductive to like non-conductive, which kind of like feels plausible, but like how do you get that into the brain? I was like, oh, nanobots that can cross the blood-brain barrier, like hand wavy. I don't know. It's like the stuff that is hand wavy, like make it like one or two sentences, and then the stuff that you're like, oh, there's like stuff like this is more plausible. You can like add the detail that makes it. I don't know. I don't know how if that was a good example slash explanation.
Yeah, no, that was a really cool example. Yeah, so I think it goes back again to balance, where it's like, again, this is not, you're not writing a research paper, so you're not expected to, nor should you literally explain every step in like the actual detail that would be needed to write a scientific paper. At the same time, I think reading like scientific research papers can be really helpful. Like if someone hasn't done research before, it can be really helpful to read the papers to see like the, I guess, scientific process, like what experiments do you start with, and like how those are built upon. And yeah, I think the point about detail is like a really good one, that definitely you can't, well, if you're writing like a hard science fiction piece, you have to explain something, but yeah, that you don't have to, explain everything. Then you can pick and choose what to explain. And um yeah, and I guess, you can always find your local biologist and run it by them if if that's a plausible thing. I think that
Yeah.
would, yeah, I think if like especially for popular media, I think if certain script writers just like ran it by a biologist, that would help avoid like the completely wrong stuff.
Oh my gosh. Yeah. Okay. So I have the one it like sort of I don't know, annoying media representation of science. Okay, I absolutely love the show Never Have I Ever, but I don't know if anyone's watched it, but I'm sorry, the lab scenes where she's basically in this she's in this lab and like the person she's like dating like brings food like takeout into the lab
Oh no.
while she's like aliquoting stuff. Nothing was labeled, absolutely nothing was labeled. I was like, does she even know? Like I don't know. It was ridiculous. It had bothered me every single time that lab was shown. I was like this is horrible. This would like not pass the EH&S inspection. This would not like this would just be shut down. Like, oh my god.
Yes, I highly empathize with the lab setups in media not being anywhere near what they should be.
Yeah, okay, like those things of like their random like ampules of glowing substance. Why aren't they
Yeah.
labeled? It wouldn't take that much to just label them.
Yeah, at least like, my rule of thumb is like if you're gonna, be wildly inaccurate, at least make it cool. So at least tell us like what's in the, vials, and that would actually help make it more accurate in this case.
People don't eat in next to like chemicals.
Oh yeah, yeah. This is the takeaway. The audience takeaway. People don't eat, yeah, next to their experiments. Like if you're doing like
They weren't
brain
PPE.
slices.
Like, they weren't wearing any PPE. They were just like, yeah, I don't
Yeah.
know. That is not.
Scientists wear gloves, they wear lab coats, they wear eye goggles, especially if they're working with something highly dangerous. Um,
Shoe covers, hair nets.
yes. Yes, so we want to see this in everyone, all the science fiction writers in the audience, we want to see mentions of PPE in your next work. Put it in there. Oh, okay, this is a good one. So, is there anything like that biosciences doesn't understand or that's like completely unknown, but that lay people would be surprised that how thoroughly we don't understand it yet?
Probably what consciousness even is from a neuroscience perspective.
Right, because basically we don't know anything about that, right?
Yeah, like I mean, I was on a panel yesterday where this was brought up, but it's yeah. And
Yeah.
honestly, even just like the scale to which we still don't know how the brain works. I like people are like, oh, uploading or simulating brains and things like that. We're pretty far from that.
Yeah. Yeah, it gives me hope as someone who's just starting to learn about the brain that I will be able to contribute because there's so much to contribute to.
Yeah.
And when going back to like the very molecular basics, I think I mentioned this briefly, but yeah, basically we don't really know what like 99% of the human genome does, and probably more than that because I mentioned there's this 1% of the human genome that encodes for like the proteins and enzymes that do stuff in the cell. But most of those genes, we also don't know what most genes actually do. Um, so I think this is yeah something people don't realize just like the depths of what we don't know. Like we don't know like what the protein the gene makes looks like. We don't know what that protein does. We don't know what would happen if it was gone or if it was like altered in any way. Um, and yeah, I think this is important for people to know because we always hear, especially like in the popular media, like, oh, I sequenced my DNA, I know what all my genes are, so now I know like all this stuff about myself, I know everything about myself, and like that, like those companies that say they do that, it cannot be true because we just don't know what like most of the genes do, and let alone the stuff that is not genes in the genome. Um, yeah, so like sometimes when I think about it as a biologist, I think like, wow, how can we possibly understand something like the brain when we don't even understand like what most of our DNA does? But on the other hand, it's like, probably to understand our DNA, we have to study like complicated structures like the brain, because if we just study, cells in a dish, like some of those genes are related to neurodevelopment, so we can't understand everything unless we study those really complicated systems. So it's really interesting because it's not like a linear process for biology where you study like the simpler thing and then you work your way up to the more complicated thing. Like you kind of have to study everything at once to like know about the basic things sometimes.
Yeah, and I mean, and in some ways we know a tremendous amount about the brain too. Like, and maybe like in the near future, we would be able to like simulate interacting systems of like memory, decision making, like like sensory perception and all sorts of stuff because we understand them at especially like a systems level. But then you get things like, for example, like um someone in a one of my friends, Jonique, her research is Alzheimer's research, and so like her like there's this sort of kind of theory that one subunit of a single receptor um like an alteration in that single receptor subunit can like end up causing like behavioral effects that are like in mice similar to like, you know, like completely like disrupting behavior and like memory and all sorts of stuff. And it's like, how do you sort of factor that in? How do you like, that's what's really like underexplored. Like we would need to know like what we can cut out, and to do that we need to like do a lot more research and understanding of like the brain at every single level before we actually try, you know, like cutting it out and making like a lower dimensional functional brain. Like we would need to know exactly what we need to keep and what we can get rid of.
Right. Yeah, I think that touches on a really important topic, which is there's like certain areas of biology where our knowledge is very deep, but where it's so far like lacking a lot is like connecting those to like other areas which have been historically thought to be historically not collaborative. Like basically we have researchers studying every single different part of the human brain, and I think only in recent years has like the collaboration had people really started appreciating like how the extent of collaboration we need between these different like subspecialties, and between the broader ones like neurobiology, immunology, like molecular biology. um so that's one thing I would like to see a lot more in science fiction, which is like emphasizing how important like collaboration is, not just for biological sciences but also science in general, which is like most research now is not done by like lone geniuses. I would also doubt the veracity of such in existence of such a thing in biology. But yeah, basically if you look at research papers now, they're done by usually like huge teams of people, because like yeah, specialties have gotten so specialized that one person cannot possibly do them all. So yeah, I'd love to see that more in speculative fiction as well. Teams of people working together. All right, so I guess we're in the last 3 minutes of our um, panel, so I guess we can start wrapping up and maybe like doing a brief outro, and then uh, if the audience has any more urgent questions, we can um, put it in chat now, and we'll be in Discord afterwards as well. So, Arul, where can people find you and stuff?
So my Twitter is at Arula Rathnakar, like my first name, my last name. And then again, all of my stories are on Clark's World magazine. And yeah, that's probably where to find me in my work.
Cool. And I'm on Twitter at the world she saw. And yeah, we're gonna be in Discord in the Houdini. Is it called a room? We're gonna be in Houdini after this. Um, and channel. Yes, we'll be in the Houdini channel.
Thanks so much.
Yeah, thank you everyone for coming. We'll be answering more questions and stuff.
Do we just leave?
Yeah, I guess we have 2 more minutes, so if someone has like a super
Oh, yeah.
like urgent question, or I guess we can. Okay, main takeaways. Please have your scientists wear PPE and be more collaborative.
Read research papers.
Read research papers. No eating in the lab. That's really important. All right, I guess we'll be in Discord. How do dreams work?
Oh man, no, you should have asked that like that. I don't know if I can explain. Okay, I mean, in
60
one
seconds.
minute,
Elevator pitch.
uh, reintegrating short-term memories into long-term memories and pruning unnecessary experiences out of your brain.
Cool. Perfect. All right. We'll see everyone in